We're just getting them to play the recorded talk. Hopefully it'll come on soon.
Good day, everyone. My name is Dalit Tivedi, and I'm a master's student at the Northwest University. Today, I'm going to be presenting to you my project titled Characterizing the Historical Suitable Habitat of Bolinas globosas in the Gombe District, Limpopo Province. Schistosomiasis is a vector and waterborne disease that is transmitted by schistosoma parasites from freshwater snails. The symptoms include abdominal pains, enlarged liver, blood in stool or urine, fever, and even diarrhea. The disease is prevalent in tropical and subtropical regions of sub-Saharan Africa. This includes South Africa, which has 4 million people infected with schistosomiasis. Figure 1 shows schistosomiasis prevalent areas, and Limpopo and Mpumalanga show the most prevalence. Schistosoma amatobium is transmitted by Bolinas globosas and is geographically distributed on the eastern side of the country. The optimum habitat preference for Bolinas globosas showed that this species prefer aquatic areas that have aquatic vegetation, water conditions that are thermally shallow, sometimes temporary and unstable water bodies. This species was also found in inland streams that are swampy and have boulders present in the water. The Bolinas globosas was also shown to thrive in fresh water at a pH range between 6.8 and 8.6. The species was also shown to survive at temperatures that are constantly high, ranging from 15.5 to 36 degrees Celsius. The precipitation levels was shown that this species prefer post-rainy seasons at a range of 600 to 900 millimeters. This study aims to characterize historical suitable habitat distribution of Bolinas globosas in the Gombe District, Limpopo Province. The Gombe District falls under the Limpopo Province, historically known to provide suitable environment for the snails. The Limpopo rural areas were found to have a prevalence of 63 to 70% of schistosomiasis. This disease affects rural areas more because of lack of good sanitation facilities, poor healthcare facilities, and lack of access to safe water. The results from the study will be useful for monitoring the potential shift in the spatial range of these intermediate host snails. Currently, limited studies have characterized the suitable habitat of schistosomiasis on a district level. Figure 3 shows a map of the Gombe district along with the rivers and occurrence data within the district. The area of the Gombe district is 25,597 kilometers squared. The climate is semi-arid that has dry spells that cause drought in the area. The maximum temperature in winter is 10 degrees Celsius and summer can have a maximum at 40 degrees Celsius. The precipitation level is 500 millimeter annual rainfall with 87.1% falling from October to March. The population is approximately 1.2 million people, with 55% of the population females and 45% males. The water source in the community make use of river water and borehole water. 7.4% of the population have access to piped water that is inside their dwellings. 16% of the population have access to flushing toilet, and 16.5% have access to waste removal weekly. The Member District has 112 clinics, 6 district hospitals, and 29 mobile services. To characterize the historic suitable habitats of Bulinas scabrossus in the Member District, the National Freshwater Snail Collection provided secondary data used for this objective. The data range from 1957 to 1963. This raw data included the plants, surface water, salinity percentage, water transparency, speed of water, substratum, water body, altitude, temperature, and rainfall when collecting samples. The effect size was used to find the importance of each variable and how much the variable contributes in the analysis power. The decision tree was used to identify patterns in the data sets. The results were in table format. The results for the Bulinas scabrossus from 1957 to 1963 showed the highest preference at permanent surface water, freshwater salinity, clear water transparency, slow running water speed, brooks water bodies, muddy substratum at an altitude of 500 to 1,000 meters, rainfall 600 to 900 millimeters, and a temperature range of 20 to 25 degrees Celsius. These are the results for the effect size, and they show that water bodies had the highest effect size in 1957 to 1963 for Bulinas scabrossus. This study identified a historical suitable habitat or using raw data from the NFSC, creating tables showing the number of times and counts as well as effect size of the snail. The study is important for monitoring the potential shift in the spatial range of the intermediate host snail. The preferred habitat for Bulinas scabrossus were brooks in permanent slow-running water with mean annual temperature range of 20 to 25 degrees Celsius and rainfall of 600 to 900 millimeters. Furthermore, suitable habitats also included clear, fresh water with the presence of aquatic plants and a muddy substrate. These are the references. Thank you.
Interesting and important, interesting talk of something really important work. Do we have any questions for Dolly? No one in the audience? Are there any questions online? Okay. Dolly, you mentioned that you're doing this work, to monitor potential shifts in the distribution of the snails. What sort of shifts would you expect? Well, what variables would you expect to cause these shifts in distribution?
I would say temperature and rainfall, especially temperature, because Biomphalaria in Bulinas scabrossus, they prefer different temperatures. So with that, we can see that the increase in temperature would result in globosus being favored, your Biomphalaria being favored, but a decrease in temperature will result in globosus.
Thank you very much.
Thank you.
Last chance for any more questions? We are all good? All right. Thank you so much, Dolly.
Thank you.
Okay, our next speaker is Tami Wanda. He's doing a talk on the geology and geomorphology of the Tukela Bank, towards the ground up marine spatial planning. It's also a recorded talk. Can we please play the talk?
Greetings to all. My name is Tami Wanda. And today I will be talking about Beneath the Waters, where we are looking at the evolution that has happened in seabed mapping. I'm sorry for not being able to join you in person, and I hope you guys will enjoy this virtual talk. So my area of interest was between Durban and Richards Bay, where we looked at mapping sites for the Smart Zone project, where 14 sites have been mapped, which have totaled to about 267 square kilometers of the seabed being mapped, which are found on different restrictive level zones for the management of the Wet'suwela banks and PA. On the right-hand side, you see planning and you see the research vessel that was used and platform for data collection in the images on the right-hand side. The aim and objectives of this study is to discuss the evolutionary advances that were made in seabed mapping, and also to show the value and importance of multi-beam echo sounders in seabed mapping, especially for marine protected areas. So looking back at history, you can see that there's been quite an improvement in mapping the seabed, where in the ancient, not ancient, in the past times, they would use lead line method, which was just basically getting single points of whatever area they were at to see the actual depth of that area below them. And that was the lead line method, which then progressed to single beam, which mapped wherever the vessel transversed or moved above the surface of the water and didn't map anything on the sides. And now we have progressed to multi-beam, which maps the entire swath of the seabed, making sure that there's overlap and the whole seabed is actually surveyed. As seen in the images, the multi-beam echo sounder uses sound to collect hundreds of thousands to millions of georeference depth soundings, which are processed to create a 3D digital terrain model of the surveyed area. This technique captures high spatial resolution data of the seafloor's geomorphology, which can be used to characterize the marine habitats in terms of their georeference location, their spatial extent, and geomorphological characteristics. And after processing this data through software such as HiPAC and QPS, this data can then be displayed in Surfer or ArcMap, for example, and one can produce output maps that are seen on the image on the right-hand side at the bottom. This only possible though if the components of the system are working in sync with one another. So recently we published a review paper which was titled The Value of Multi-Beam Bathymetry in Marine Spatial Planning for South Africa, which looked at the value of how multi-beam can be applied in marine spatial initiatives within the country. Not only does it show that, but this paper shows the contribution that multi-beam echo sounders are to make in seabed mapping, where we, in our results, compared our results to the earlier data acquisition that was already available of the seabed. We can see that in the left-hand side image indicated with the yellow block, the area on the Wet'suwela area of shores and Kwazi, which is represented by both inset A and inset B. This, looking at both these data sets, you can already see the differences in detail and the resolution of the features on the seafloor. So this is what our aim was, to actually bring more information towards marine spatial planning, so that better decisions can be made in terms of spatial extent and spatial distribution of the seabed features. I do not have time to go through all the findings of my results for each individual area. However, I would like to just briefly discuss the overall spatial distribution of the 14 surveyed sites, which total to about 267, which we revealed from that that 96.5 of that was covered by reef area, And then 57.5 of that was covered by bed forms, and the remaining 113 was covered by flat sediments or featureless seabed area. From our results of our study, we show that with the technological advances made within our field, well documentation of the seafloor can now be achieved with multi-beam echo sounders. And because of this, multi-beam echo sounders can then be used for both short-term and long-term monitoring of the seabed, which then in turn helps monitoring the habitats that are found existing on it. We believe that mapping will ensure improved application of conservation, sustainable use, and management of the biodiversity for equitable benefits towards socio-economic stakeholders and the public within the appropriate scale. And because of the variation observed in these structural characteristics of the seafloor in these preserved reef complexes, we believe that this is indication of possible number of biological niches for different species that are compatible for different areas or environment. And knowing these habitats in detail and knowing their environmental extents will improve our understanding of the interaction of the existing ecosystems. And lastly, mapping of MPAs and these polar shorelines quantifies the interplay between sea level fluctuation and geological settings of those polar shorelines, which is something that interests the geoscientific community, because then we are able to then predict what the future holds if provided the coastline was to sea level was to rise again, given that we are in a state of global warming. In closing, I would like to say that not only will this data be important to the geoscientific community, but it will also form the baseline data for other research studies in other fields such as biology and ecology. And with that, I would like to say thank you.
Thanks so much, Tammy. That talk was very interesting. And you guys, I think, do have some of the coolest toys to do your work with. All right, any questions from the audience? Okay, we have one at the back there.
Yeah, thanks, Tammy. Yeah, exciting stuff that you guys are doing up there. We are interested in knowing how low sea levels and connection of adjacent river systems might have influenced the distribution of freshwater fishes, for example. And I'm just looking at the work that you are doing and looking at how far are we from perhaps reconstructing the Paleo River drainages within KZN, for example, which is quite close to where you are, because it can really help us to explain the distribution patterns and the genetic patterns that we're finding for some of the freshwater fishes from that region. Thank you.
To my understanding, I'm not much educated in terms of the fishes and freshwater, since we mapped so far offshore. Yes, there should be some interaction with the freshwater and the marine, given that there are rivers feeding into the marine space. But I had not looked at it in that sense. My main goal was actually mapping the habitats in the marine space, then later on to try and see which species are actually there, then try and link them with and see how then that habitat is accommodating for that ecosystem. And then that is only when I would be able to see what kind of species are we finding, actually just marine species, or is there also freshwater species within the area. But given that I have a geological background and not a biological background, that is something that is yet to come in the future, as this is still work from my ongoing PhD.
Thanks, Tammy. Thank you.
Yeah, thanks very much. Can you explain to me, just in simple layman's terms, the difference between the data that you get from a multi-beam compared to a side scan sonar, which is the way we used to do it? How are they different? Is the multi-beam more data rich? Is it better?
To my understanding, the side scan sonar didn't have elevation. Or am I mixing the systems of SSS side scan sonar? It didn't have elevation to it. It showed the flat surface because, if I remember properly, you had to drag it with a rope behind the ship or the vessel to actually try and get it as close as possible to the seabed to actually see what's going on at the bottom. Whereas with the multi-beam, it is capable of measuring depth, and also it does this all the way from the sailing on the top of the water. I'm not sure if that answers the entire question.
Sorry. Thank you so much, Tommy. Have a lovely day further.
Okay, thanks.
All right, before I announce the next speaker, I'd like to just make some announcements. First of all, we'd like to do a group photo at lunchtime. So please, everyone gather around lunchtime for a group photo. And then also there's a silver gray polo that's lights are on. So if you have a silver gray polo and your license plate is LH31 NWGP, your lights are on. Please go turn them off. Nobody. Okay. Alrighty then. All right, our next one is also an online one. So Tabani's giving a talk on, mapping historical schistosomiasis, risk areas in Johannesburg and Tshwane municipalities.
Greetings, everyone. My name is Tabani Kwela, and I'm doing my master's at the Northwest University. Today, I'm going to be presenting a study that maps the SAIAB schistosomia risk areas in Johannesburg and Tshwane municipalities. But very briefly, I'm going to start by explaining what schistosomiasis is to sort of give everyone an idea of what I'm talking about in the next slides. Schistosomiasis is a waterborne disease that is caused by the parasitic flatworms known as schistosomes, and it occurs mostly in the tropics. It is also regarded as the most neglected disease in sub-Saharan Africa. An infection rate of 60 to 80% has been reported among school-going children in poor communities in South Africa, which makes children the most vulnerable group. And about 5.2 million people are thought to be affected in South Africa. And there's a range of factors that sort of contribute to an increase in schistosomiasis infections in endemic areas. These include poor access to clean water, sanitation, hygiene practices, as shown here in figure 2, where we have children playing, swimming or bathing in essentially infested water bodies. Other factors include age, gender, and access to water. Figure 1 basically shows us the areas that are at risk to schistosomiasis in South Africa, and we can see that this disease is mostly concentrated in the eastern parts of the country, also extending into the northern regions, which includes Limpopo and also some parts of Gauteng. There are specific freshwater snails that are required in order for this disease to be transmitted to a human host. The ones that are common in tropical Africa includes Biophyllary fiveri, Bulinus africanus, and Bulinus globosus. The occurrence of schistosomiasis in South Africa has been linked mostly with the prevalence of Schistosoma hematobium and Schistosoma mansoni. So these freshwater snails are highly sensitive to fluctuations in temperatures, and that can be attributed to their poikilothermic nature, which means they respond very rapidly to sudden changes in climatic conditions. Other factors such as rainfall, floods, droughts affects the snail's distribution, reproduction, fecundity, and its survival rate. So this table basically shows us the habitat requirements for each species, And we can see here, for example, for Bienfalara, we have this species requires a temperature range of 22.8 to 27 degrees Celsius, whereas on the other hand, Linus africanus requires a temperature range of 0 to 8 degrees Celsius. And we can argue that this species requires or prefers relatively cooler temperatures compared to Bienfalara 5.3. The remaining factors are similar for both species. So what is the aim of this study? This study aims to identify the areas that are vulnerable to schistomiasis in Johannesburg and Tshwane municipalities. And the objective of this study is to map the vulnerable zones in Johannesburg and Tshwane municipalities. Why is this study important? This study is important because mapping schistomiasis risk areas at a microgeographical scale is essential, as most infections occur at a local scale, as the local people are most likely to be exposed to the transmission site or the possibilities that contain the infected cells. And also not forgetting the fact that these people rely on these water bodies for other domestic and recreational activities. We can also argue that risk area mapping is a useful tool for showing the areas that are at risk to schistomiasis, and it also ensures that the resources are allocated to the populations that are at risk, and that can help a schistomiasis control efforts. So this slide basically shows us a study area map and also the characteristics of both municipalities. So we have indicators of vulnerability, which we've used in this study. This includes exposure factors and also susceptibility factors, which I will look at in the next slide. But for exposure factors, we have total population and land cover, and also there's temperature and precipitation. So the susceptibility factors, they do like really contribute to the, the distribution of this disease in endemic areas. So this diagram basically shows us the methodology that we have followed in order to obtain the results for this objective, which is to identify the areas that are vulnerable in Tshwane and Johannesburg municipality. So here we've got the results that shows the areas that are at risk to schistomiasis infections as transmitted by Bienfalara 5.3 in the Tshwane municipality. And we can see that Irene is one of the areas that is characterized by very high. Here we have another map that we have generated that shows the areas that are at risk to schistomiasis as transmitted by Linus africanus in the Tshwane municipality. And really we do expect to see some differences in the maps as the species prefer different environmental conditions. And as a result, we can see that some parts of Upper Richville are characterized with a very high risk to schistomiasis infections. The following areas, Johannesburg South, Soweto, and Randburg, were characterized by a high to a very high risk to schistomiasis infections.
Here are some of the important points to ponder. So we have observed the following areas to be characterized by a very high risk of schistomiasis infections, and also there are parts of that were characterized by very low risk to schistomiasis in the Tshwane municipality. On the other hand, we had the following areas which had very high vulnerability to schistomiasis infections, and also there were parts in the Johannesburg municipalities that were characterized by very low risk to schistomiasis. And also these results are important because they serve as an approach to improve schistomiasis control efforts, and this will undoubtedly help identify target population to be characterized for action, thus minimizing the costs. And also, historical mapping of schistosomiasis is, will help pave the way for future studies and thus prevent outbreaks of schistosomiasis in Gauteng province. And with that being said, I'd like to thank you all for listening.
Sorry. Hi, Tabani. Thank you so much for that. Again, very interesting talk on some really important work. Does anyone have questions for Tabani?
Hi, Tabani, Francesca here. Thank you for your talk. I was looking at the characteristic of the species linked to the precipitation. I wonder if some of your areas that you're targeting are also affected maybe by drought, and if you have seen any variation, or if you think the drought could affect the distribution of the species, of the two species, because they were equally influenced by the same levels of precipitation. Thank you.
Okay, thank you for that question. Well, actually, that is true. So we do have, cases where this disease or the infected snails, are being affected by the droughts in these areas, these two municipalities, as actually the reading material that I've looked at that have shown that droughts really do occur in these areas. I'm not sure if that answers your question.
Tabani, anyone else have any questions?
Thank you, Tabani. Lalia. I just want to ask, how well does your risk model translate into real world numbers?
That is actually a very interesting question. I'm not sure if I do have an answer for that for you today, yeah. But I'm actually, looking to see, if whether this, the method that I use, weighted overlay analysis, is really supposed or predicts what it's supposed to be predicting. I'm not sure if that answers.
Hi, Tabani. I'm just wondering whether you also explored some seasonal dynamics associated with the risks of schistosomiasis in general. I surely want to be interested in knowing when likely the risk of schistosomiasis will be high.
Can you please? I didn't get the question.
Did you manage to explore some seasonal dynamics associated with the risks of schistosomiasis? Season, the effect of season.
Tabani is asking about seasonal dynamics. Did you detect any seasonal dynamics?
Pardon?
Seasonal dynamics, season.
Oh yeah, that's something that I actually looked at on my second objective. This is one of the objectives that I'm looking at for my dissertation.
Thanks so much, Tabani. Okay, our last online speaker for this session is Enya. She's measuring the effectiveness of marine protected areas for commercial line fishing sector in KwaZulu-Natal.
Good morning, everyone, and thanks for taking the time to listen to me talk about the multidimensional impacts of marine protected areas in KwaZulu-Natal on the commercial line fishing sector. My name is Enya Manting, and I'm a master's student at the Department of Environmental Science at Rhodes University. So to introduce you to the project, the first thing that I thought was important is to introduce you to the commercial line fishing sector. These commercial fishers are not trawling or doing long lining, they are in fact fishing with hooks and lines, either on fishing rods or hand lines from boats such as the one in the picture on the left. It's the oldest fishing sector in the country, being traced back locally to the 1500s, and is economically also very important as it creates the most employment of all fisheries sectors and pays the third highest salaries and wages. In KwaZulu-Natal, 52 vessels are allowed to operate with a maximum of 354 crew in any year. Currently, only 30 of these rights are active. These fishers rely on catching smaller reef species when they are most abundant. Fishing of all types remains the biggest cause of marine biodiversity degradation, as well as the greatest pressure on marine biodiversity, despite mounting pressures from other sectors such as oil and gas and mining, according to the National Biodiversity Assessment of 2018. As you can see here, KwaZulu-Natal is an important area for the commercial line fishing sector. In 2020, with the declaration of new marine protected areas, all of the country's largest coastal marine protected areas were declared along the KwaZulu-Natal coastline, making this an interesting area for understanding the types of tensions that exist between people and marine protected areas. So this brings me to my project, the aim of which is to build and contribute knowledge on the efficacy of MPAs in KwaZulu-Natal for the commercial offshore line fishery by using a multi-methods approach to determine firstly how MPAs impact commercially important fish stocks, and secondly how the MPA network and its current governance affect commercial line fishers. So my project had both an ecological and a social component, and for the ecological component, we used baited remote underwater stereo video systems to compare commercially important fish populations inside and outside marine protected areas and in various management zones. For the social research component, I held structured and semi-structured interviews with 22 of the 30 commercial line fishers active in KwaZulu-Natal to understand their perceptions of MPA effectiveness, the livelihood impacts of MPAs, and the governance of MPAs. My project is part of a 3-year-long project called the Smart Zone Project, which aims to collect baseline data for a larger 5-year project called the Utikela Marine Protected Area Ecosystem-Based Adaptation Project. So on the top left, you can see a BROV in action, and on the bottom left, event measure software being used to measure fish for maturity and biomass calculations, which were important for this project. On the right, you can see pictures of KwaZulu-Natal and its marine protected areas, which from north to south are Isimangaliso, Utikela Banks, Olive Shoal, and Protea Banks marine protected areas. And in the Eastern Cape, there's Ponderland Marine Protected Area, which is still relevant to this project since it is relevant to the fishery. The red squares indicate the rough location of the fishers who I managed to speak to, and the dots, which are now indicated in purple, refer to the sites which we actually used, which were on mesophotic reefs between a depth of 30 and 130 meters. So let's look at the results. Well, if we look at the commercially important fish species only, it seems that ecologically speaking, marine protected areas are doing their job. This holds true for both abundance metrics and biomass metrics. When we look a little bit more deeply into the situation, we find that it really is the restricted zones inside the marine protected areas that are doing all the work of protecting fish stocks. Again, this is true both for abundance metrics as well as biomass metrics. Moving on to the social component, the first question that I had for fishers was whether or not they believe it's important to protect and manage fish stocks. Fortunately, 100% of the fishers believe that this is important. So I asked them next whether they believe that MPAs are a good tool for doing this. And interestingly, the majority of them believe that yes, it is. So the next question I had for them was whether or not they believe that their community supports MPAs. And interestingly, the majority of fishers again indicated that they do support MPAs. However, about a quarter of them indicated that they believe their community actually opposes MPAs. And so the question begged to be asked, well, why? Is it because MPAs impact your relationship to the sea? Fortunately not. So I moved on to perceptions of fairness. Most fishers do not believe that the MPA impacts are fairly distributed along the KZN coastline, and neither do they believe that the enforcement of MPA rules is fair. They also don't believe that their rights are taken into account in MPA planning and management, and neither their livelihood needs. When asked about whether or not there was any process that they could follow in case problems arise in relation to the management of marine protected areas, the majority of fishers said that there was no such process in place, whereas a few of them cited an expensive and time-consuming legal process which had recently come to a close. So my next question for them was, I knew full well that an extensive stakeholder engagement process had already occurred prior to the MPA expansion, but I needed to find out from them what this consultation process looked like and whether or not they felt that whether it existed in their minds and whether they felt that it had been meaningful. Interestingly, about a quarter of the fishers indicated that there had been no consultation process, meaning that the stakeholder engagement process had not adequately reached all the fishers who it needed to reach, while the majority of the rest of them believed that this engagement process constituted mainly only an opportunity to comment on things that were already going to happen, but not that their consultation had made any sort of meaningful difference to what occurs. And so here, in trying to find a solution to the problem, I like to defer to my education in the social sciences, where I realized how important language really is. So I decided to look at the language we use when we talk about how we include people in decision-making processes. Engagement, stakeholder engagement, really is, according to the dictionary, about creating a feeling of connectedness, and therefore is actually an inadequate way to discuss the way we do this. However, in South Africa, we don't really use this terminology. We actually talk about consultation. However, consultation as well is only a meeting which is held to discuss something. Although it gives more space for everyone to talk, it doesn't necessarily mean what we need it to mean. So if we're looking for the correct word, I'd like to suggest that potentially we should move towards consultation processes, where there is cooperation aimed at effecting a unified proposal or concerted action. Perhaps it is time for a paradigm shift in the language we use about our engagement processes in South Africa to start talking about concertation rather than consultation. Thank you for coming to my talk, and I'm sorry for going slightly over time.
Thanks so much, Enya. That was a great talk. Does anyone have any questions for Enya? No. Oh, there we go. We have one at the back.
Hi, Enya, Judy here. Thanks for a great presentation and interesting results from chatting to all those fishermen, all that time ago. If you have to look at the overall perspectives of the fishermen that you chatted to, do you have an indication from them as to their idea of solutions? So what were the solutions that they came up with is my one question. And then looking at the definition of your new word, and I love the fact that you're like engaging with the consultation, etc. The definition involves opposing forces. Do you think that that perhaps puts us against each other at the very start of the process? And maybe we can find an even more exciting word to use. So just two sides to that question. What are the fishermen's solutions to you? And then let's explore words together. Thank you.
Hi, Judy. Yeah, thanks so much, firstly, to Judy and Bruce for all that they did to help my project. So the fishermen mostly had an idea of moving marine protected areas, which they spoke about quite often. But I also got a solution from them, which I think most people would agree on, and that's that marine protected areas just need better enforcement, even as they currently are. So that was quite promising. And yeah, I'd love to explore words together. I actually really enjoy these processes. So I think, yeah, it's definitely better not to put ourselves against each other from the beginning. So I'd be open to other suggestions, of course, but I thought at least to start with from the premise of specifically trying to create a solution together, you know, I think that, and yeah, the aim of consultation is to create something together. So whether we start from opposite viewpoints, I think it's relevant and perhaps contained within that as well. Yeah, thank you. I hope that answers your question.
Great, thanks, Enya. Is there, okay, there's one more question for you.
Hi, Enya. I'm quite intrigued by the lexicon that you've just introduced. But on a serious note, is it not just a matter of semantics? Is it not just a case of where we need to do more action rather than play around with words?
Yes, of course. I mean, my project really only received a full set of ecological results very recently, and I'm still very much only starting with my thesis writing. So in actually finding real solutions, I'm planning to do a lot more work on that. But I thought a good place to start is with our semantics, because our semantics do set an intention for what we're trying to do. And, you know, at this early stage, I can't really speak too confidently on how to change the current situation. I know the change is necessary, but I feel like I need to do more homework before I can step into that conversation. So I thought to start with semantics. Thank you.
Super, thanks so much, Enya, for that. So our next talk is in person. We have Nobuhle doing a talk on light trap efficiency validation for larval assemblages in urban coastal habitats.
Good morning, everyone. My name is Nobuhle Mpanza. So today I am going to be giving a presentation on a pilot study that I did, which feeds into my PhD. Firstly, I'd like to just introduce urban coastal environments, which is the focus of my actual PhD. So we have two situations currently. We have natural rocky shores, which are highly complex in structure. If you're looking at that picture further on my right, they're highly complex in structure, they're biological hotspots. Because of this complexity, they house a number of different species, which allows them to interact, allows feeding, allows a number of important ecological functions. They're also very important, especially for the early life stages, which is the focus of my PhD. But then we have a current problem, which is urbanization. We have a situation where those natural coastal environments are being changed or transformed into concrete hard structures for creation of harbors, housing, etc. And this means that we're losing these important biological hotspots or biological habitats for different species. So for us to better understand, in terms of understanding the community structure, it's crucial. Understanding community structure is important for conservation efforts, especially within urbanized environments. And then since I'm focusing on the larvae, which are the early life stages, those are the ones who actually tell us how the population structure is going to look like. So in terms of sampling the larvae, you need to select a certain tool that works. So this pilot study for me was trying to validate the efficiency of two designs that I had at my disposal. And also because sometimes what you're finding working in natural rocky shores might not be working in urban sites, and the other way around, what works at urban sites might not actually be as efficient in natural rocky shores. So I wanted to test whether different designs of light traps can be deployed at either the natural sites and the urban sites. And also I wanted to test their efficiency in terms of collecting the actual larvae. These are my study sites along the east coast of South Africa. So I had 2 large harbors, which was Port of Ngqura and Port of Port Elizabeth. I also had 2 natural sites, which was Kinney Bay and Cannon Rocks. And then I had 2 small harbors, which was Port Alfred Marina and Port of St. Francis. So I was very fortunate that when I started my PhD, there was already 2 designs that our team, the course team, had at their disposal. But for me to choose the best tool for my specific research, I needed to do prior field-based research just to understand how they work. So how a light trap works is it's deployed in the evenings so that it works throughout the night. It targets positively phototactic species. So when it's nighttime, the light is on, of course, and everything that's positively phototactic comes into the lights. So another factor that I needed to take into consideration was when you're deploying them, they need to be at least 5 meters apart because you're trying to avoid interference between the intensity of the light between the 2 light traps. So I had to place them 5 meters apart along my seawalls. The 2 designs that I had a quatrefoil, which was taken from Patrick, which is my course supervisor, Patrick. And then I also had another design, which was a much cheaper one, cylindrical, which was firstly developed by Chen et al. And then Seshni, which is our SAIAB MSc student, kind of improved on the design that already existed. So I had 2 designs at my disposal. So the quatrefoil one is a more expensive design and it's more robust. It's made of HDP, DPE, and it's a light trap alone, just one structure is 9K, whereas the other one is way cheaper and it's easy to make and it's made from water, 5-liter water plastic bottle. So since I'm working in urban sites and natural sites, it's very important to take into consideration, especially if you're working with such an expensive equipment, because you don't want to deploy your light trap, your expensive light trap in the natural rocky shores. The following morning you come, it's not there. It's been stolen or it's been washed away. So those are the factors that I was trying to look into so that I know which design I'm using in the urban sites as well as in the natural rocky shores. So when I was looking at there's a very preliminary results based on just what we're finding so far. We looked at the diversity that we're finding between the 2 designs. And then we found that there is no significant differences when it comes to the actual species diversity. And this is mainly because it's within the same marine bioregion in the eastern coast. So you're expecting that you're finding more or less the similar species. So there was no significant differences in species diversity. Although when you're looking at the larger harbors, which is Ngqura and Port of Port Elizabeth, you're finding that the quatrefold design, which is the more expensive one, was functioning more better than the cylindrical. And also what other things that you're observing was that in Ngqura kind of acts like a natural site in terms of the natural environment conditions. So we're finding that when you come the following morning to collect your light traps, the quatrefoil one is still there, it's intact, and you're getting your sample. But then when you're looking at the cylindrical, which is much cheaper, it's either squashed, washed away, or it's just upside down and you're not finding anything. So this was just our baseline in terms of taking what you're finding in Ngqura, which imitates natural sites, and trying to see whether we're continuing with the natural sites or not. So I also looked at their species, abundance and richness, and then I found significant differences. And then we found that the quatrefoil again had higher species richness and abundance. When you're looking at species richness and abundance, you found that the quatrefoil was performing way better compared to the cylindrical. And then we found that in the small harbor, Port Alfred Marina, it had higher species richness compared to all the other ones, all the other sites. So what does this mean? So this was a very important step, especially for my PhD research, in terms of choosing the right tool and also deciding whether, and also trying to see if I can use the expensive one, I can risk and use the expensive design at the natural sites or not, or I just use both, either one, if there are no differences between the 2. But also another thing that we realized from this study was that you cannot deploy both designs at the same time. So it's possible that you might deploy the cheaper one, but then you're also risking the factor of you're actually going to lose your samples. They're not going to be as efficient as you're looking at. because of what happened in Ngokga. So going forward with my PhD research, the decision that we made based on our in-field observations and the actual results that we're finding so far, which we're still processing, is that the quadrifold, more expensive design is the best option going forward for my research. In conclusion, I'd like to thank everyone involved, especially the harbors, which have given us access to their property, and the course team, as well as SAIAB for funding my research. Thank you.
Thanks so much, Nobuhle. That was a very interesting talk and some really important to test your methods before you use them. I think we have some time for 2 questions. Anyone? Okay. Can we get a mic down here, please? Yeah, but Taryn, front.
It's like Rosalind said, it's nice to test the methods before you use them. Just out of interest, because I know nothing about light traps, where in the water column or where in your sites do you put them, and do you deploy it for like a whole tidal cycle, or are you wanting it? Are you deploying on the low and getting the high and back down to the low? Because obviously you got to do it at night as well with the light. So just out of interest.
So what happens is usually we work early in the morning during low tides, because we work with tides if you're the coastal environment. So we deploy in the morning. Our batteries last 24 hours. Yeah. So we deploy in the morning. Let's say I'm coming today at 8 a.m. I'm going to leave them there throughout the day, but they're not going to work throughout the day. They're only going to start being effective when it's nighttime, because there's no lights. The only light source, it's the lights in the light trap. So they start working throughout the day. So from dusk until dawn, they're more effective. And then I come the following morning, the following day, and then I take them out.
And then just a follow-up question in terms of the man-made places, so your Port Alfred Marina and St. Francis, is there some kind of influence of the ambient light that you're getting from houses or whatever may be in the area? Because obviously even the ports to a large degree, because they've also got ships and lights at the harbor. So are you seeing some kind of influence of the ambient light relative to Kini Bay and Cannon Rocks? I think you mentioned that I think in the SAIAB talk or the DIFS talk that you took those sites out, but just what's the influence of the potential ambient temperature for larval recruitment?
Temperature, lights.
I mean, sorry, lights.
Okay, so I haven't really looked specific at that, but I can say, I think, especially in St. Francis. So St. Francis, we have a lot of boats that have lights themselves, and they're fishing. So we're finding that between the cylindrical and quadrifold, there's not really difference in terms of what they're getting, in terms of the numbers. So it's possible that what we're seeing there is basically because of there's other sources of light. So it might be possible that outside lights is playing a factor into what we're finding, but on the other sides, there's not really much, especially in Ngokga. There's no other sources close. There's no other source of lights closer to where we work, because the ship are further away from our sites. But if I'm using St. Francis as an example, it's possible that the lights plays a factor.
All right, cool. Thank you so much. All right, our next speaker is Timbani, whose talk is on a multi-method approach to assess habitat use and connectivity by juvenile coastal fishes in St. Francis Bay seascape. Just quickly, while Timbani is going up, has anyone found a blue Redmi phone? Someone's misplaced their phone. So if you found this phone, please do bring it to us so that we can return it to the owner. Thank you so much. Take it away.
Good day, everyone. I'm Timbani, and I'm going to be briefly presenting my PhD proposal on a multi-method approach to assess habitat use and connectivity by juvenile coastal fishes in the St. Francis Bay seascape. Nursery habitat for juvenile fish have a high abundance and richness of juveniles. They contribute to high growth and survival rate. They do that through the provision of food and shelter. As a result, structurally complex habitats are particularly important as nurseries, as they do provide shelter and more room for food attachment. Nursery is a spatially explicit seascape consisting of multiple mosaics of habitat patches that are functionally connected. The habitat core area within the seascape is characterized by hotspots of juvenile fish, of high juvenile fish abundance. So nursery habitats that contribute to adult population more than the others are considered more important as nursery habitats. But the problem is the direct contribution of nursery habitats to adult fish population is a concept that is very difficult to actually prove or to quantify. It's not only the habitat forming species that define the seascape, but it's also defined by other abiotic parameters that play an essential role in shaping a nursery seascape. Those include depth, exposure, which is wave exposure, and seascape structure. In my PhD, we are aiming to use a seascape approach to determine nursery habitat usage by juvenile fish assemblages in an estuarine to marine seascape in St. Francis Bay in the Eastern Cape, and how the seascape context configuration influences the nursery role of different shallow nursery habitats within that seascapes. We will also try to quantify the connectivity from nursery habitats to adult habitats or to deeper waters. We will do that through characterizing the seascape through mapping and classifying benthic habitats in the shallow water nursery potential habitats. And then we will also determine the core nursery areas, which is juvenile density hotspots within the St. Francis Bay estuarine marine seascape as a function of habitat type, depth, seascape configuration, and wave exposure, and assessing connectivity between nursery and adult habitat using RAVs and BRAVs. And then lastly, we will assess the connectivity between juvenile and adult habitats within the St. Francis Bay coastal seascape using otolith and islands compound-specific stable isotopes. Our study area or our seascape, like I've said before, it's in the St. Francis Bay. We will have sites ranging from the sea point all the way to Aston Bay, but it will also include the lower reaches of Chrome, which are dominated by Zostra capensis. And then we will also move further deep for adult fishes. In terms of the method, to achieve our first objective, we will create habitat maps of both the nearshore marine environment and lower reaches of Chrome estuary. And then we will assess the diversity and cover of habitat forming structures around sampling sites. That will be your seagrass, and then the diversity part is mostly in the nearshore marine environment. And then to do that, we will take digital photographs of the substrate will be taken using a GoPro camera mounted on a PVC frame with a 0.5 times 0.5 meter quadrant base. And then we will try to quantify wave exposure. So for wave exposure around the marine sampling sites will be indirectly quantified using the gypsum method. So in the gypsum method, we kind of put the gypsum block over time, and then, we measure them before we put them in, and then we leave them there for overnight. When we take them back, we reweigh them to see the effect of wave speed. And then we will be using the ministerial RAVs and BRAVs to assess nursery habitat used by juvenile fish and connectivity between nursery habitats and adult habitats. So we have these ministerial RAVs on your left that we specially designed to sample juvenile fish. They have the length between the camera lenses a bit smaller than the ones that you used to here, that end use. And then we will deploy those for 60 minutes in the shallow, in the potential nursery habitats both in the seagrass and the nearshore marine environment. And then the data that we get there, we will analyze it using event measure, getting variables like max N, the species richness, and also species richness and length. We will also do behavior for the necessary habitats using Boris, just to see what behavioral activities that the fish are doing. Is it the ones that are associated more with habitat use, or they just swimming past? And then for adult habitats, we're also planning to deploy BRAVs, ANTS BRAV, and then we will compare the two communities and see if there is any potential connectivity between the habitats. And then the last part for compound-specific stable isotopes, different tissues actually store what we eat, and they have different turnovers. So we try to get the tissues that can keep what the fish eat for longer. And also we try to get the ones that actually grow as the fish is growing. And then the best ones are otolith and the islands. The otolith adds rings that you can actually narrow down to days. And then the islands, they keep adding layers as they grow. So the aim is when you go closer to the core of the otolith, that's when the fish was still a juvenile. The same thing with the islands. If you go narrow going to the core of the islands, that's where they were still juvenile. So the aim is to sample those and compare those to the otolith and the islands of the juveniles. If there are any similarities, that may suggest that at some point they may have used the same habitat, the adults have used the same habitats that juveniles are currently using. So for that, a core from each sub-adult and adult otolith representing the first year of growth will be isolated and removed for analysis following methods by McMahon. And then the core and otolith, the core and juvenile otoliths will be ground to fine powder. The same thing with islands. And then the powdered samples will be acid hydrolyzed, then derivatized before amino acids. Carbon and nitrogen isotopes are analyzed. Thank you.
Thanks, Tembani. You're cutting it fine there. I think we have time maybe for one quick question. Okay, we have one at the back there. Please take a mic up. And then any other questions, you can catch Tembani at lunchtime and have a chat.
Dry and there's no rain and the estuary mouth is closed. So what happens to the fish that use the estuaries as their necessary habitats? How does that impact their abundances?
I'm not really sure, as I've only worked with the permanently open estuaries, but I will assume that it depends what species you're looking at, what time do they actually spawn and what time do they actually move up the estuary. So if it's those that are actually like spawning around spring and summer, then they wouldn't have that problem. And most of them are spawning. Most of them have a lot of juveniles right now from the scoping work that I've been doing. So I don't think they will have that problem, but I'm not sure.
Well, thanks so much, Tembani. Do catch him at lunchtime if you have more questions. Our next talk, so there's a change in the program. The next talk is an online talk by Mohammed. It's entitled Trajectories of Change in South Africa's Freshwater Fish Fauna Using Historical Data to Support Assessment and Management of Threatened Species.
Good afternoon, everyone. My name is Mohammed Kaji. I am a PhD student at the University of Cape Town, and today I'll be presenting on some aspects of my PhD research, specifically looking at trajectories of change in South Africa's freshwater fish fauna. So to give you a little bit of background into the topic, as I'm sure many of you are aware, South Africa's freshwater fishes are regarded or categorized as the country's most threatened species group. So our country's unique freshwater biodiversity is under immense pressure from climate change, from human activities such as the damming of catchments and land use change, and then very importantly, facing huge pressure from invasive species such as bass and carp and bluegill. In light of this, my project aimed to collect and consolidate historic occurrence records of freshwater fishes occurring in South Africa, and then to use these records to assess how have these freshwater fish populations been changing over time. The ultimate overarching aim here is to provide a comprehensive update to the state of knowledge of freshwater fishes in the country. So data collection for this project started way back in 2018, whilst I was still a master's student. We searched for occurrence data for all freshwater fishes that were known to occur in South Africa. Our data sources come from a wide pool, including peer-reviewed published articles from Google Scholar. We've included the SAIAB entire fish database, research-grade data from GBIF, and then we also embarked on a years-long mission to try to get private experts and government organizations that had unpublished data to share those data with us so that we could publish them. All of these data are now available on our online platform called the FBIS, or the Freshwater Biodiversity Information System, where we have more than 60,000 freshwater fish records publicly available. The very first paper from my PhD was just a description of the data set. So this was published in the Journal of Limnology. And in addition to just describing the data set, we were also able to publish our data on the GBIF platform. So it's now currently housed on 2 platforms publicly available. Firstly, the FBIS, and then secondly on GBIF. Diving into the data set, we find that the data varies both spatially and temporally. From a temporal perspective, we have 195 years of data, starting from 1828 all the way to this year. But we find that data are not equally distributed. So 89% of data are collected after 1975. Interestingly, we also start to observe a slight decrease in the numbers of records in the last decade or so. And so it's interesting to dive into why that might be the case, whether we've stopped sampling enough, whether government funding for monitoring programs has decreased, or whether it's just a product of the fact that we haven't had people upload their data or done as thorough search in the more recent years. From a spatial perspective, we also find that the data are unequally distributed. Converting the actual occurrence records into a density map, so these are the number of records per quarter degree square, we find that there's a high concentration in the number of records in the northeast of the country, but also along the south coast and in the western parts of the country. The interior parts of the country seem data scarce. And this could be because we aren't sampling in these areas, but also, as we are well aware, many of these river systems in the interior of the country do not contain that many fish. And in some situations, actually, the rivers dry up completely for many months in the year. The other output that was associated with my PhD was to develop a master species list of freshwater fishes occurring in South Africa. So prior to my project, there was no comprehensive species list in the country. We've then since then updated the National Biodiversity Assessment List, updated the definition of what exactly is a freshwater fish, and our final list now contains 134 species on it. This is obviously an iterative process, and I'm sure if Albert is in the room listening in, he knows that very early next year, we're going to start bothering him again to try to help us to update this list and then get it published so that we've got one comprehensive list of all of the freshwater species or all of the freshwater fishes that occurs in South Africa. Our current list contains 129 species with records in South Africa. So 105 of these are native and 24 are non-native. And then we've got 5 species that are on our list that do not have data. But after consulting with experts, we were told that these species definitely do occur in the country. We just did not have digitized records for them yet. Importantly here, I want to note that we did not include any undescribed or yet to be described taxa. Additionally, we also produce these species lists at provincial level. So it's important from a management perspective that our provincial conservation authorities have information on what species they have in each part of the country, but very importantly, what are the non-native species and what are the threatened species. The second paper to come out of my PhD is hot off the press, was literally published yesterday, and this looks at spatial distribution of species in South Africa. So the first thing that we did is just looking at species richness across the country. We find species hotspots in the northeastern parts, and then very small hotspots in the western parts of the country. And then very importantly, when we look at endemic and threatened species, we find hotspots in the Cape Fold ecoregion. Concerningly, we see that the non-native species distribution seems to mimic that of the threatened species distributions. Next, we wanted to look at how effective the protected area network in South Africa was at protecting South Africa's threatened freshwater fishes. And what we found was that 57% of records for threatened species occur within a protected area. And this translates to roughly 36% of the threatened distribution range overlapping with the protected area. Concerningly, when we look at the overlap between threatened species and non-native species, as you can see in figure D, we found that 58% of the distribution of threatened species was also occupied by non-native species. So there's a strong overlap between threatened species and non-native species in South Africa. Similarly, when we look at the distribution of non-native species in relation to the protected area network, we find that almost 47% of South Africa's protected areas have non-native species occurring in them. And then lastly, the last paper that I am going to talk about for in this discussion looks at how did we use the data that we've collected to contribute to conservation and decision-making at a national level. So what we find, or what we found in our previous paper and what has been published quite extensively, is that South Africa's protected area network does not do an adequate job at protecting South Africa's freshwater fishes. In response to this, we've collaborated with the Department of Forestry, Fisheries and Environment through the DFFE screening tool to contribute fish data to the screening tool to ensure that fish species are included in this piece of legislation. So what the screening tool does is it means that any new development needs to be screened for environmental sensitivity. And now through the work that we've done, we're able to include freshwater species into that process. So how does this national environmental screening tool work? We've taken the FBIS data and handed it over to the screening tool by developing current distribution maps as well as species distribution maps in order to flag potential habitat where threatened freshwater fishes occur. How does this work visually? So for example, for Barnard's rock catfish, what we've done is we've taken the occurrence record, put it through a species distribution model to develop an accurate species distribution for the species. And that then gets fed into the screening tool so that you've got the current records, the model distribution, the predicted habitat, and the current known distribution based on the records, which then goes into a GIS layer, allowing for the species to be protected under a national legislature. So in this regard, we were able to develop these final maps for threatened species occurring in South Africa. On the top left, you've got the medium sensitivity layer based on species distribution models. On the bottom left, you've got the current distribution based on occurrence records for threatened species. And then on your top right, you have just the critically endangered species occurring in South Africa. And through this process, we were able to provide additional protection for almost 49,000 kilometers worth of river system. Okay, in conclusion, I just want to highlight the importance of the freshwater biodiversity information system, as well as the importance of digitizing our data and making it publicly available. And then lastly, the importance of collaboration. Everything I've presented today comes out of huge amounts of collaboration across the country with experts from around the country to try to get these products publicly available, but also in a format that can be used by conservation managers. Thank you very much for listening.
Thanks so much, Mohammed. What an important contribution. Wow. We have time for one quick question. Does anyone have a question? No? Nobody? Okay. Oh, okay, here we go. Sorry.
Hi, yeah, great talk. Always cool to understand things spatially and on maps. I was wondering if there's any tool or whether it'd be possible to almost filter that down just into what species exist in each catchment. So, for example, each independent riverine catchment almost have like a list of the species that are found there in a supporting document. Because sometimes it's quite nice to be able to just, know that, okay, within this river's catchment, there are the species. Because often it's done the other way, where it's the species and then the catchments they found in. Which, I mean, they're almost exactly the same, but sometimes researchers are not so much interested in the fish, interested in the catchment, but then the fish information is important, if you know what I mean. It just might be quite useful, but I don't want to give you any more work.
Yeah, thank you for that question. I'm a little bit scared to answer it because the reality is that our species list and the data are not entirely cleaned yet, and we've realized with the paper we've just published that actually erroneous data still exists in our data sets, and cleaning it is a process that's probably going to take a couple of years. So in the paper we've just published, and I can share that in the comments, we do have a supplementary material that's got all of the provinces and all of the catchments at primary catchment level. And we've provided species lists for each of those at each of those spatial extents. What we then realize is actually there are erroneous data that means that some species are included in certain catchments where they definitely don't exist. So what needs to happen next is a thorough cleaning of the data. And that process is going to take a while because obviously, I mean, we don't have the expertise in just my supervisory team. So we need to then meet with experts for each catchment and go through each of our species lists and determine what's there and what isn't there and what should be there that isn't. So yeah, it's in the pipeline. I think it's definitely something that will fall onto my plate once I submit my thesis. For my PhD, we're trying to submit this coming February, so it's quite a tight deadline, and we're submitting it as is. But I think the next steps would be to, exactly like you say, publish species lists and even distribution maps at catchment level. I know that I saw a paper and I've forgotten the title of it. I know that Josie South and them had published a recent paper. It might have been on the Kawi catchment or the Chorits, I'm not sure, where they've done exactly that. They've given a kind of breakdown of the entire catchment. And I think it's important from a management perspective that we start sending out these outputs at usable scale. So I fully agree. I think it's something that needs to happen. But hopefully it falls under some sort of postdoc that I take on next year.
Great, thank you so much.
Cool, thank you very much.
All right, our next speaker is Mpilo, whose talk is on assessing day-night changes in shallow and deep water temperate reef fish assemblages using environmental DNA metabarcoding and SAIAB.
Good morning, everyone. As Ross said, I'm Mpilo, and I'll be presenting on investigating day-night changes in fish assemblages on shallow and deep temperate reefs using environmental DNA and SAIAB. So basically, apart from being economically and socially important, fish play a major role in the marine environment, and understanding the interaction between fish species and their environment is important for conservation and management. So a lot of factors affect the structure and diversity of fish species in the marine environment, including depth and the dive cycle, which is a pattern that is observed in a 24-hour period, and it's known to influence fish behavior and activity, which leads to the changes in composition and structure in fish assemblages. So even though this is the case, most studies limit or are restricted to shallow water sampling and only diurnal sampling, probably because of practical difficulties associated with sampling during the night and also in deeper depth zones, and also the logistical challenges involved in selecting the appropriate sampling methodologies. So when assessing fish assemblages, studies have used extractive and non-extractive methods. Most studies have used underwater visual sensors methods, probably because of the minimal impact to the environment and to also the target organisms. Over the past years, baited remote underwater stereo video systems have been shown to be an effective sampling technique to assess fish assemblages on both temporal and spatial scales across a range of different habitats. Stereo browsers provide opportunities to limit some of the biases that is posed by UVC, such as accurate estimates of lengths and also sampling in deeper depth zones, which is restricted by scuba diving when it comes to UVCs. Over the years, recently, a invasive sampling technique that is rapidly developing to address ecological questions in the marine environment is environmental DNA. And this environmental DNA, compared to traditional monitoring technique, has been shown to effectively detect higher species richness. However, even though this is the case, it's used, despite the demonstrated effectiveness of environmental DNA as a tool for assessing and detecting fish species, its use for evaluating day-night changes in shallow and deep reefs remains unexplored. Which then brings me to the aim of my study, which was to compare environmental DNA metabarcoding and stereographs to investigate day-night changes in fish assemblages on shallow and deep temperate reefs. Specifically, we aimed to investigate how the fish assemblage structure and diversity between time and day and depth zone differed, and whether the patterns observed were consistent between the two sampling methods. So to do this, we sampled at 2 locations, namely the Tegama National Park Marine Protected Area and at Ryebanks in Algoa Bay. And our sampling design consisted of paired eDNA and stereograph sampling in the shallow and in the deep reefs. And in the shallow reefs, we sampled during the day and again at night, and did the same for the deep reefs. So basically, this is just a process of environmental DNA. We filtered 4 liters of water per site, and then did the DNA extraction, amplified the DNA using 16S primers, and sequenced the samples using Illumina-MySeq sequence. And to filter and process the samples, the sequences, we used the mother software. And then for stereographs, which was deployed immediately after collecting water samples, we used Event Measure to identify the fish and also provide mix-in. So for statistical analyses, we fitted generalized additive mixed models to test the effect of location, time of day, depth zone, and methodology on total species. And then we used we did our community analysis on presence absence data on primer. So basically, this is just a Venn diagram which showed us, which was plotted for environmental DNA and graph data for day and night. And basically, it showed us that environmental DNA detected much more species than stereographs, and also showed us that it basically detected more unique species that were observed in the environmental DNA data compared to the stereograph data, yes. So our models, basically the not equal sign shows that it's significantly different, and the equal sign showed, yes, significantly different and not significantly different, yes. So basically we see for species richness that environmental DNA obtain higher total species or species richness compared to graphs in the deep. And then this pattern, the pattern observed from day to night, the trend is basically similar for eDNA and graphs in the deep zone. Yes, in the deep zone, and not so much in the shallow, yes. And then when we look at the shallow versus deep trend in species richness during the day only, we see that there's a significant decline in species richness for stereographs, but not so much for environmental DNA. And in the same trend, but during the night only, there is a significant increase in species richness for environmental DNA, but not so much for stereographs, yes. So then our Permanova community analysis was significant for all the factors, including the interactions. And then we plotted an MDS, which didn't show clear clustering of samples, but showed the variability within the environmental DNA data compared to the stereographs data. We then proceeded to plot the CAP analysis, which showed distinct clustering of methods and also depth zone, the factor, significant factor of depth zone, but not so much for time of day. And we see that there's different communities basically depicting these changes, and stereographs were more correlated to shy sharks and the Black Sea catfish, while environmental DNA was dragonets, which was not seen in stereographs, and shallow water hake. So overall, environmental DNA detected more species compared to stereographs, and also detected more unique species. And as I said, for example, the dragonets, it's species that are cryptic, because most species that were detected by eDNA, in addition to the species that we saw on browse, were very cryptic, so we're not seen on browse videos. Both eDNA and stereographs showed a general decrease in total number of species detected in the day compared to the night in the shallow depth zone, and an opposite trend was observed in the deep depth zones. The significant pattern of the patterns in species richness across the environmental gradients varied by method. And lastly, community analysis showed variability within environmental DNA data compared to stereographs data, and both eDNA and stereographs showed an effect of method and depth zone on the community structure. Yeah, thanks.
Cool, thanks, Mpilo. Unfortunately, we're running out of time, so please catch Mpilo at lunchtime with your questions. Sorry. Sorry. Okay, so our next speaker is Thembelihle, whose talk is on building the foundation for efficient monitoring of mesophotic fishes to support management of the Utukela Banks marine protected areas.
Greetings, everyone. My name is Thembelihle Dube. So the study site for this research is the Utukela Banks marine protected area that is located in KwaZulu-Natal between Richards Bay and Durban. Both commercial and recreational fishing is known to be taking place in the mesophotic reefs of the MPA, which are offshore historically important fishing sites. And these fishing activities pose threats to mesophotic fish diversity, and therefore long-term monitoring is very essential in order to support management. Okay, sorry about that. In order to support management of the MPA, and for long-term monitoring programs to be effective, they need to be ecologically robust and also need to be achievable with available infrastructure as well as financial resources, which then brings us to the aim of the study, which was to develop an optimal LTM program in order to support management of the Utukela Banks marine protected area. And the objectives were to determine the effect of environmental variables, depth, benthic habitat, depth, as well as relief influence on fish assembly structure, and also to develop a baseline assessment of fish populations in the marine protected area among different zones of the MPA. And then the focus of this presentation is on the third objective, which was to determine the optimal sampling requirements for an LTM program. So for our methods, we used multi-beam ecosounder in order to survey key reef sites of the MPA. Following that, the data obtained from multi-beam ecosounder was used to classify reef relief and randomly allocate samples in GIS. Following that, we conducted stereograph surveys using the baited remote underwater video systems, which were selected or chosen because they are very robust, cost-effective, and also non-extractive tool. All statistical analyses were conducted in R, where we did our generalized additive models. So our general results show that there's no significant difference in species richness and abundance between the reef habitats, but significantly significantly lower species richness in sand habitats. And we also saw that there's both species richness and abundance decreased with increase in depth. And then this plot here, the main pattern that was observed is that in the high profile reef, the restricted zone had significantly lower abundance than both controlled zones, but the abundances were comparable between the restricted and the exploited sites. So following that, we did the power analysis in order to consider a sampling size necessary in order to detect changes in fish assemblage structures through time. The species sketched for it's for slinger. The reason why we selected the species is because it was the most abundant species, and also it's the important species for fisheries in the MPA, and also because of its because it is of higher conservation priority. So what we see from here is that we'll need a total of 36 samples in order to be able to detect change, and in order to be able to detect 10% change of the sling abundance with a statistical power of 80%. We then did the cost benefits analysis in order to measure the benefits associated with monitoring of the MPA. So doing a cost, the cost per BRAF sample will be this much, meaning that we'll need 1 day in order to analyze videos. And then the cost per the entire reefs will be 36 work days. So what we did was to identify and map 13 reefs. Following, some of the reefs were joined, giving us a total of 11 reefs and two reef complexes per management zone, plus 1 reef in the outside the MPA. So our alternative scenarios, if we consider doing a full survey of the entire reefs, in terms of representation, in terms of completeness, it will be a true representation of fish assemblages occurring in the area. However, this is unfeasible for both time and finances. And then reducing a survey to only doing 5 reefs will mean the reduced sample and therefore less time spent analyzing videos. And doing a partial survey, in terms of completeness, it is a bit compromised. However, in terms of time, it is good as it's approximately, it's gonna take about 6 months. However, the finances will depend on the availability of financial resources. An example for monitoring schedule for Tugela banks will be in a 16-month process, whereby in the first month we collect data, and then for 12 months, we analyze data, do video annotation and data management, and then for 3 months we do reporting. This process could be repeated once every 3 to 5 years. And thinking from a management perspective, for example, the SMVelo KZN Wildlife will consider monitoring the MPAs. For instance, they could do monitoring of 2 MPAs per year, combining the Tugela banks with Aliwal Shoal and Ismangaliso and Protea banks in that 1 year, and then consecutively they could do, in consecutive years, they could then take a 2-year break from field work where videos are analyzed. And this process could be repeated maybe after 5 years. However, because the management usually don't have big teams as well as massive budgets, monitoring biannually or annually might not be feasible. In conclusion, we saw that there was more abundance in in reef than in sand habitats, meaning that spatial comparisons will need to be incorporated. And stirrup show they have a high statistical power. However, future studies will need to consider methods of optimizing the post-field data. Thank you.
Okay, thanks so much, Timberlike. Unfortunately, again, we don't have time for questions. Please do catch her at lunchtime though, and do engage. Okay, our next speaker is Sipendulwe, whose talk is on automating anomaly detection in telemetry dataset and fish movement patterns using long, short-term memory and autoencoders, a time series analysis approach.
Okay, hi everyone. I am Zaza Sipendulwe, and I will be talking about my project, my MSc project. So basically, we are automating the process of anomaly detection in telemetry data. So our objective is to come up with a machine learning model that will be able to predict anomalies and to be able to automate anomalies in telemetry. So the first question that you will ask yourself is, why are we interested in telemetry data? So the simple answer to that is we are interested in telemetry data because we want to understand the aquatic animal behavior and their movement in natural habitats. And we know that aquatic data has been used worldwide and it's used for studying the movement of aquatic life, and this method helps us it allows us to gain information on the movement of the fish at different levels, from large scale nationwide migration to fine scale migration. One of the questions that you will ask yourself is, what are the problems with telemetry data? So we know that researchers usually aim to gather a lot of data when they are conducting any study that they are conducting. So with that now, mistakes happen, especially if you're working with data set that contains a lot of data that contains maybe hundreds of thousands of detections to millions of detections. So this issue becomes even more if you are trying to study a small habitat where there are a lot of animals that are tagged. And the problem with tagging a lot of animals in a small restricted space like an estuary is the creation of false detections and anomalies. And those anomalies can be in the form of environmental factors or technological limitations. So in our data set, we have 50 dusky corp were tagged, and their movement was monitored by 16 receivers that were deployed in the bread estuary between 2016 and 2021, and together together these fish amassed more than 3 million detections. So in our data, there were various variables which include fish ID, the receiver, and the station, the latitude and the longitude of the station, and the corresponding date and time of the detection. And we also have the distance. So in order for us to be able to identify these anomalies, we have three criteria that we created. The first one is if an individual fish stayed at only one station for the period of the study, so we regard that as an anomaly, as it suggests that the fish may be deceased, as we can see on the graph that it stayed on only station 2 for the whole period of the study. So the second one is if the fish is moving as you would expect it to be, but suddenly stays at one location for more than 120 days, so we also regard that as an anomaly. So the third criteria is that if the fish is moving down the estuary as you would expect it to be, but suddenly it is recorded kilometers away in a receiver that does not make any logical sense. So we regard that as an anomaly. So if we have a look at our graph, as time goes on, we can see that the fish is moving up and down the estuary and it's being recorded consecutively. But here now on this part, okay, here now we can see that it's detected as station 1, but now it moves to station 5. So this is an anomalous behavior, because in an estuary, the receivers are placed in such a way that a fish cannot pass consecutive station without being detected. Hence, we are saying that this is an anomalous behavior. So this is basically the flowchart that we have created based on our criteria. So this basically summarizes everything in terms of how did we find our anomalies, and this is the distribution of the anomalies in the data, and we can see that the data that we're working with is very unbalanced. So this is our method that we're using, which is the LSTM autoencoder. So firstly, before we input the data into our LSTM autoencoder, so what we do, we clean the data, we remove the noise, and we try to scale the data. So what this does, so the LSTM encoder takes in the LSTM encoder takes in the sequence data, and what it does, this LSTM autoencoder learns to understand and capture the patterns of the data, and then after capturing the patterns of the data, it compresses the data into a compressed version, which is Z, and then Z now is taken to the decoder. The decoder, what it does, it tries to reconstruct the input of. It tries to reconstruct the input, which is the sequence that we put in here from the compressed version. So now, how do we identify anomalies? So firstly, we train our model on normal data points, and then from there we try to find the threshold, which is the limit. And then after that, what we do, we calculate the errors, which is the reconstruction error. So basically, the reconstruction error is the difference between the input and the reconstructed input. So if the error is greater than the threshold, then we are saying that this is an anomaly, and then if it's less, then this is a normal data point. So in terms of the results, these are not the final results, but I can say maybe they are preliminary results, because there's still a lot of work that needs to be done, because the results are only coming from the basic LSTM autoencoder. Our aim is to come up with a model, with an LSTM autoencoder model combined with the generative artificial neural network to come up with models that will be able to predict anomalies. So if we have a look here, okay, so we have the confusion matrix for the balanced data and the imbalanced data. So basically what it does, it shows us how many true normal that we got from the unbalanced data and how many true normal that we got from the balanced data. So we can move on here so that we can, this will explain a bit more on that. So on table 1, we have accuracy, F1 score, recall, and precision. So for the unbalanced data, we can see that the accuracy of our model is it's 60, and for the balanced data, it's 96. So the accuracy on its own, we cannot say that our, let's say for example, on the balanced data, we cannot say that we have 96% accuracy, then our model is doing well. So the accuracy on its own does not tell us that our model is actually doing well. So if we go to table 2, then we can see that we have unbalanced and balanced data. So there is data detected as, okay, just 1 minute. Okay, there's normal data detected as normal, which is for the unbalanced one. That means our model is correctly identifying that 98% of the time, and then there's normal data detected as an anomaly. So, which means our model is misclassifying 2% of the normal data as an anomaly. And also we have anomaly data detected as an anomaly, which is our model is correctly identifying that 22% of the time, and then it misclassifies 77% of the time, which is a very big problem. So now what we did, we tried to balance the data so that we can have better results. So if we can see on the balanced side, we can see that on the normal side, it misclassified only 0.26% of the time, which is an improvement from the from the unbalanced one. And then normal data detected as an anomaly, it does that correctly 62% of the time. And then we have anomaly detected as normal, which is misclassification, which is 37%. So basically, these results are not final, but yeah, they're not final results. So what we are aiming to do is to combine this LSTM autoencoder with a generative adversarial neural network to come up with a model that will be able to decrease the misclassification, so that we can have a model that will be able to identify anomalies in the telemetry data. Thank you.
Thank you so much. Sorry, unfortunately, we don't have time for questions. You used up your question time. Our next speaker is Sivu, whose talk is on seascape mapping and modeling, shallow water habitat mapping in Cape Recife, Algoa Bay. Please do catch the students at lunchtime if you have questions, and the online audience, if you send us your questions, we'll get the answer for you.
Good morning. It's me, Sivu. My project, my MSc, is basically looking at mapping and modeling the coast of Cape Recife. So seascape ecology is defined as an emerging science that applies the concepts of landscape ecology to the marine environment. It usually helps us describe the effect of seascape structure on the biological assemblages that we might find in the habitat. So for example, we would look at the area, the type, quality, spatial arrangement of the habitat, and look at how it will affect the abundance and distribution, life strategies of these organisms. In my project, I'll be mainly looking at seascape structure, which encompasses the composition and configuration of the habitat. So I'll be looking at number of patches, patch sizes and type, and also referring to the spatial arrangement and shape of the habitat. And then the effect of seascape structure on biological assemblages could be determined using habitat mapping. So effective mapping of the seascape is crucial in order for us to develop a robust understanding of the seascape structure. It also provides us baseline spatial data for marine spatial planning, monitoring, sampling, and conservation efforts. When mapping, we usually use methods like acoustic or visual imagery using sonar systems or manned equipment. So examples of those is multi-beam, echo sounders, and drop camera systems. So I aim to combine and interpret multi-beam data and seabed images to map and classify the benthic habitat of the western corner of Algoa Bay. We'll do this by classifying the substratum type and biological benthic components of the study area using photographs, and then also classify and map the habitat based on seabed characteristics, bathymetry, and biota using the multi-beam bathymetry, drop camera images. So my study site is Cape Recife, which is influenced by the Algoa Current. It is mainly made up of sandstone rock platforms with incised pools and gullies. So in the nearshore area, we usually find microalgae, which is the Plecanium chlorhiza. But then as we move more offshore, we're not really sure what kind of microalgae or biota we may find. So in order to achieve the first objective, I'll be using a drop camera system in order to get images of the benthic habitat. Once I've received my images, I will identify the sessile and mobile invertebrates and the substratum type using Katami as classification, and then also trying to find the abundance of the benthic invertebrates using CPCE. For my second objective, I'll be looking at multi-beam data and then it will be processed and cleaned so that I can get data and generate maps using benthic terrain modeler on ArcGIS. Once I have my bathymetry data, I will combine the data I got from my drop camera images and the bathymetry in order to create a holistic habitat classification map. Thank you.
Oh, short and sweet. Alrighty, does anyone have some questions? Mpilo, I mean, Sibu, sorry. Got Mpilo on the brain apparently.
Just a quick question. Why Cape Recife? Why did you choose that area? Does it have anything to do with the proximity of the little MPA in that area or something like that?
Not as such, it's just part of the bigger project that I'm in, which is the Seascape project. So I was given that site to map and model.
Sorry, okay. Any more questions from the audience? Okay. Alrighty, thank you so much, Sibu. Oh, great. Our last talk is from Dana, movement behavior and migration patterns of flathead mullet, Mughal cephalus, in the Kawi Estuary, South Africa.
Hi, everyone. My name is Dina Mukari, and I'll be presenting my PhD, preliminary results of my PhD. It's not done. Yeah. So on the movement, behavior, and migration patterns of flathead mallets, and this is part of a bigger project as well on the mallets in the Kawi Estuary, South Africa. I'm supervised by Dr. Taryn Marie, Josie Peg, and Prof. Ember. Just to give a little bit of background about estuaries, as this is what my project is based in. Estuaries are defined geographically as areas where the sea and the freshwater meet. And then due to this, they are very productive because they receive nutrients from both water bodies. And then they have high species diversity. And then which means increased ecological function. And they're also necessary ground for marine species. So estuaries are also good as they have goods and services that they provide to the people and food, for example, and recreational opportunities, but they're also threatened by overexploitation, habitat alterations, and which can lead to loss of connectivity. I mean, especially since I mentioned that the connectivity between the sea and the freshwater. So the species that I'm studying is in the family Mucilidae, which is small fisheries that are mostly found in estuaries, and then they go to the sea to spawn. And these species are economically important, aquaculture species in some countries, even commercial, and they are also important to the society. People use them as bait as well as for recreational and even food. So Mughal cephalus is also known as flathead, is the most distributed of the family. There's a map there showing occurrence data from JBIF, which is where you can find it across the world. So it can grow up to one meter. Well, yeah, and then it matures around 35 centimeters. And this species has been known to have a bigger habitat compared to other mallets. Yeah, so other mallet species that can be limited to, oh God. Sorry. Other mallet species that can be limited to, some limited to the sea, some in certain parts of the estuary. This one has been shown to have a bigger range going all the way from the river to the estuarine environment, to the sea, closer to the sea. So just to give a little bit of information that's available on flatheads from across the world, since the species is cosmopolitan, It's been known as a resident or migratory population, can be divided into resident and migratory population in some countries. And then in some parts of the world has been shown to do partial migration, and some individuals even have irregular movement between the sea, the estuary, and the fresh water. And then in some parts, it spends the whole life in the marine environment. And then in South Africa, in places such as the Bushman's estuary, they've been known to go all the way 100 kilometers from the sea. So they go into the fresh water part. And then in St. Lucia, this was an interesting study done recently whereby when the St. Lucia estuary mouth closed, the flathead were actually decreasing in numbers because they couldn't reach the sea to go spawn. Which leads me to my aim and objective. So my whole entire PhD is on flatheads, and then I'm planning to look at the movement of flathead between the freshwater estuary to the sea. And then I'm also planning to look at the migration history, which can be found using otolith microchemistry. And then at the same time, I'm also planning to look at the thermal tolerance of mallets, as I might have forgot to mention that they are highly tolerant to salinity and temperature. So I'm planning to do this in the lab to see what's their tolerance. But then for the purpose of this presentation, I'll be focusing on the movement, and then I'll be using acoustic telemetry and then to assess the connectivity of the species between the freshwater estuarine and coastal habitat, and then link this movement behavior to environmental parameters, salinity, temperature, tidal, etc. So this is the study site in Kawi, and I've divided this into lower reaches, middle reaches, and upper reaches. But then Kawi is 21 kilometer estuary, and then each receiver has been placed 1 kilometer from each other. And then, so from receiver 6 going to the mouth, those are the lower reaches, and then going upper, that's the middle. And then the upper reaches of the estuaries from around 18 kilometers from the mouth all the way to the river. And then, so we morphologically identified the flatheads. Unlike the other species, which are too similar, flatheads are very distinct in their adipose eye and their flathead. And then we identify them morphologically, put receivers in the system. Okay, I'll skip more on the methods because I think telemetry has been explained a few times due to time. To go into my, so we take 21 flatheads, and that's their size range and where we take them in the estuary. And then this is some of the preliminary results, which shows that from receiver 1 all the way to receiver 21, these are the detections at each receivers. So as you can see, flatheads are more, they are using the whole estuary all the way to the riverine system. And then this is individual movement of each fish up to the 21st fish. So they are going all the way from the river, which is the yellow one, and then the sea mouth or the mouth of the estuary is number 1, which is blue. So they are going up and down in the estuary. And then this is some more detailed individual movement showing them doing up and down. But then it's also important to note that we didn't download all the receivers here. So some of them are missing. This is why this is not a complete presentation of the result. But what to take from here is that they do move up and down throughout the estuary, all the way to the river and to the mouth. And then some of the ones that had 1 dot or 2, these are the species that we think they might not have made it. But then mallets are also very much prey species, so they might have been taken by other species, or even some are recaptured and then they're sold in the market, some of them. And then it's something else to note is that there's illegal gill net fishing that occurs in the estuaries that are targeting some mallets. So that could be the reason for some of them missing. And then Just a quick conclusion. So mallets are successful because they have dietary flexibility and they also have tolerance to salinity and temperature. And then they also have intraspecific variation, which means that other individuals can behave in a different way. So there's partial migration, there's irregular movement, but not the studies have been done in detail in South Africa, which is where the study comes in. But mallets are also prey species and are highly exploited because they occur in a very wide range. So this could also highlight the importance of protected areas or close seasons for the species to prevail. And then again, this is just that diagram showing their habitat range. Thank you. And I would like to thank my supervisors and the fishing team involved in making this project possible.
Thanks so much, Dina. It was a very good talk. Unfortunately, because you use your time up, we don't have time for questions. Please do catch Dina at lunchtime or online audience, send us those questions. We're going to hand over now for another Flickr session. But
while we're getting ready for the Flickr sessions, can I just ask, as soon as we go to lunch, please get to the amphitheater outside if it's not raining, so we can take a photo, a group photo. Thank you.
Okay, now let's see if you are listening. So there's no need for a demonstration this time. We'll just get straight into it. So the first question is, what does the abbreviation MPA? Can you? Oh. Okay. What does the abbreviation MPA stand for? A, Marine Policy Area. B, Marine Protected Area. C, Master of Public Administration. D, Music Publishers Association.
Please make sure your hands are not on the diagram.
So everyone's answer has been captured?
Yeah.
The correct answer is... the correct answer is B, Marine Protected Area. So everyone got... Whoop.
No.
I am judging that person. So most of you guys got the answer correctly. Next question for you. Name the student that tested the efficiency of quadrifoil and cylindrical light traps. A, Tamsan Mawanda. B, Tabani Kwela. C, Nobuhle Mpanza. D, Enya Manting. Yeah. Judy and Bruce. Oh yeah. Oh, sorry. Judy and Bruce. The correct answer is C, Nobuhle Mpanza. So this time everyone got it right. Third question, this is a waterborne disease caused by infection with freshwater parasitic worm. A, schistosomiasis, B, cholera, C, that, and D, that. Schizo something, yeah.
Your fingers from
The correct answer is A, something, yeah. Next, next question for you. Oh, a few people got it wrong. Fourth question, more than 4 million people in South Africa are infected by, and it's a cause of 200,000 deaths per year in sub-Saharan Africa. A, true, B, false, C, maybe. Flash. Yeah. The correct answer is A, true. Most of you didn't get it correct. Next question.
last.
This is the last question for the clicker session. This has been fun. Name the student that has a study combining eDNA and cerebrobs to study day-night changes in shallow and deep temperate reef fish assemblages. That's a mouthful. A, Tembelihle Dube, B, Muhammad Kaji, C, Tembani Mkhize, D, Mpilontle Nyawo. The correct answer is D, Mpilontle Nyawo. And most of you did get that one correct. Thank you so much.
Hi, online attendees. Can everybody turn on their video, please, so that we can just take a photo?
Robert, you can mute. You can mute them then.
Great. There's still a few people who are a bit shy. It's just 2 seconds, please, everyone. Just a few more people that we're waiting. Ryan, are you gonna take the photo?
Cool. Are we ready to go?
I think so. I think these are most of everyone who's willing to take a photo. Okay, is everyone ready? Big smiles and 3, 2, 1.
Yeah, hold on.
Cool. Did you get the photo right?
Yeah, got it.
Great. Thanks everyone. Enjoy
Right.
lunch and we'll be back at 2.